Ladies and gentlemen, I'll speak about an algorithm for the analysis of evolutionary relationship, and so comparing different sequences. It's based on Klein 4 groups. Phylogenetics is the study of ancestral relationships among biological sequences, and sequence analysis is often performed. These are presented as phylogenetic trees, where the branches of the tree reflect the evolutionary relatedness between the sequences. We will show how it works for a Klein 4 group algorithm, which may be used in the analysis of relational databases of nucleotide or amino acid sequences. Such databases are based on the relational model of data, and we recently showed that standard genetic code table can be shown in that can be represented by that model. We have a set. First, we have a set of operators of 4 elements: E, A, B, and C. Identity, transition, transversion, and complementarity. And those operators will show how they generate distance matrices concerning Cayley table positions, row positions, and column positions. It will be tested on a data set of REC proteins in bacteria, eukaryotes, and archaea. It's REC-A in bacteria and RAD51 in eukaryotes and RAD-A homolog in archaea. So basically they are the same proteins, they're functional homologs found in those species, and they're essential for the repair and maintenance of DNA. Due to their importance and the fact that without them, basically, the repair system is endangered, and we wanted to see how it works in all 3 kingdoms. With respect to matrices, similarity or dissimilarity matrices can have application in sequence alignment and phylogenetic analysis. So it will be done, as I said, in bacteria, archaea, and eukaryota. We present it on 22 organisms from these domains of life. We have, because more sequences, if we use, I can have 40 or 45, the patterns remain the same, and the trees are very difficult to follow in one sample. So basically, REC-A is a 38 kilodalton protein. As I said, it's essential for the repair and maintenance of DNA in bacteria, and it has been found in every species and serves as a sort of archetype for this class of homologous DNA repair proteins, RAD-A and RAD-A. So here you see how it works when the DNA unwinds and has errors emerge or breaks and so on. REC-A and his homologs bind and repair it. So it is a sort of question one may ask: who emerged before? But because in bacteria, for example, without it, you can't have repair. And it's that famous SOS signal that basically makes living and repairing of DNA during replication possible. But let's put aside the philosophy and those questions. Pure genetic analysis will be done with, in the classic way, using multiple sequence alignment, muscle software. And the second step with Mr. Bias algorithm, using default options with UniPro software. So there are several models which deal with analysis of phylogenetic tree and evolution. The classic Hughes-Cantor model, as you can see later, they all resemble square of oppositions and biosemiotic square, which is rather interesting. The only thing that differs is that in Hughes-Cantor model, each base sequence has an equal chance of changing. So transitions, transversions, and transversions, which are actually complementarity, because you changed ring and group, have all equal chances. Another Kimura two-parameter model gives a possibility of different substitution rates. And Kimura also made the three-parameter models, which I showed yesterday. If you turn it for 90 degrees clockwise, you actually obtain the pattern of Aristotelian square of opposition or probabilistic square of opposition. So, just a short notice, neutral theory of molecular evolution holds that most changes occur at the molecular level, and that those slight neutral changes shape the evolution. This concept of non-Darwinian evolution, which are mostly due to neutral mutations and genetic drift, was also addressed by King and Jukes in their famous paper. So Kimura, King and Jukes actually started this type of concept of a new neutral theory of molecular evolution. The Klein group representation of mathematical operators, I showed it yesterday. Basically, you have U, C, G, and A. U as a starting point because it has smallest ring and less reactive group and so on. Is a two-dimensional arrangement of 16 boxes in which we can put the third base as individual amino acid. It can be represented in digital form, in numerical form. Permutations can be done concerning E and C, and A and B, so it means complementary. The whole table is actually a XOR algorithm which separates strong and weak hydrogen bonds. Actually, that resembles the biological situation. And it's interesting to notice that this representation, so a sort of decoded standard genetic code table from the gray code to normal regular form, when nothing changes except G and A column, unfortunately in 3 dimensions. So there are some switches at the first base, at the second base, and at third bases. in that context, we obtain rumors. So black fields are not dependent on a third base, and white fields do depend. Of course, A group sums partly additional information on and then draws of, for example, protein degradation, and so which is situated in this column. It's interesting that so through all 4 letter tables, U, C, A, and G, this is invariant. Now, I also showed this slide tomorrow. I will repeat it for the ones who hasn't been here, so that they could track. So the coded on the square are rings, and groups according to standard chemical nomenclature, which are most distant from the sugar. So those rings, and those are main rings that constitute in making Watson-Crick and Hooke's-Steen, they are invariant in Watson-Crick and Hooke's-Steen bonds. And we have 4 operations. So these operators are U, and then while the system evolves, we have an operation of transition, transversion, and transition-transversion. So Klein model for group is basically a good model for the situation we have in genetic code. So we are lucky to have a classic piece of group theory from the end of 19th century, which was partly addressed from the point of, for example, truth functions, and Marquand's PhD done, the mentor was Pierce. So this clustering into 2 classes and then another 2 classes, so Pierce was his supervisor, and the thesis was published by the end of 19th century. So those 3 several squares, biosemiotic, probabilistic square of opposition, the calculations can be made on the unit interval, and of course Kimura. Now I have to hurry a bit because Massimo Di Giulio addressed some time ago, oops, sorry. So in 2001, one problem, and that is the problem of tautology. And he said, the title says most of it, the origin of genetic code cannot be studied using measurements based on PAM metrics because it reflects the code. He's partly right. He was actually criticizing the Knight simulations. In 2004, we published a paper regarding this, when we had the same or very similar feeling about the problem of using PAM metrics, for example, to test other codes. So we randomized the data set. So we were doing investigation of different randomization of the data sets of non-homologous protein structures to see how the prediction goes. I won't get into details, but I will only say that the problem also is, at that time neither Di Giulio nor we were aware that clustering in respect to the first base, so that ambiguity UPA code Y, U excluded, is practically present in all substitution matrices as a general rule. And for the quadripeptides, for all the structures of well-defined or essentially defined helix and strands, so initial groups 4, 5, 6 and more, this cluster emerges when you scale, for example, Blossom matrix, but as well when you scale into two clusters, Mirzawa-Jerningham potential, so statistical potential, and for example, Grantham formula for evolutionary formula for polarity. Those are classic results for race, and as well as the Julius polarity score. It also works well, and atom depth. It emerges in most of the matrices and situations because it is simply a distance when you exclude ground value with respect to other operations done through Cayley's tables. So we introduced the relational model to solve some representation problem in a way that could be tuned to the relational databases. So let's go to initial Crick table. It's one of paper. It's one of the most cited papers. The presentation of the table is well known, and it's very intuitive. It gives you an idea in a very clear way what's going on, and the fact that columns represent polarity or polar and non-polar. U is non-polar, A and polar, and C and G are approximately neutral. It's easy to follow it, as well as, for example, metabolic pathways from the first G column. So the table is practical. However, as I said, it's written in gray code, and considering a relational model of the data, it is an unnormalized form. Unnormalized form does not necessarily mean that the table is bad. It simply means that it's written in a certain way. According to code introduced relational databases back in at the beginning of the 70s with SQL, those databases are routinely used. The table defines rows and columns, define fields, and some normalization of the tables should be done. Of course, it doesn't necessarily mean that unnormalized tables are not to be used. The only thing is that in some storage and manipulation with data, this helps. And so when decoded, C and G in 3 dimensions switch places, and the normalization can be performed. As I said, non-relational databases are also used nowadays, no SQL databases, but it's out of the scope of this. So in relational model, unnormalized are considered the starting point. It shouldn't be confused with the denormalization, when the normalization is done afterwards. As I said, it can be analyzed in tables as well as tables which are not normalized. And it's important to say that sometimes a joint column is considered to be not practical, but in the case of the genetic code, we have a very a very nice joint column which goes through all 4 tables and which is very important, and it's column C. So the C column is the only column which is identical in all 4 tables, and it is one, of course, joint column is one of the necessary things for the relational model to work on. So first normalized form, we can second normalized form. Those are standard things, and I don't go into detail. Eliminate transitive dependencies. So all the columns should depend on the primary key. This also. Sorry. In the end, we have first base represents row, second base column, third and first base table, and we have where, select, and form in SQL. So position of each amino acid is well defined in each of 16 boxes. Some boxes have more than one, of course. That's invariant rumor, which this invariant part, black, is actually, they mostly consist of early amino acids, evolutionary early amino acids. So due to this reason, we also analyzed the REC-A clustering in 3 kingdoms with respect to rumor as well. I will skip all those things and go. Yes, there is also one point which is interesting. Clustering into these 4 basic tables by relational model gives you an information about average number of transfer RNAs, because UNC tables have the same set, GNA tables have separate sets. So 16, 3 times 16 minus 3 stop codons gives that magic number of approximately 45. As I mentioned yesterday, relational databases can ease sequence similarity searching and large-scale genomic analysis. It can be, of course, relational model is top-down reverse engineering. It remains a question whether genetic code evolved from the, for example, 4 times 16 doublets, or it was a continuous and fast process of a recombination which spontaneously evolved. It would be speculative to try to conclude which one. As far as we know, is that this model shows in a nice way reduction of ambiguity, how the process worked, and enable us to easily follow the situation from the databases and concerning physical-chemical parameters and that wobble base we heard earlier. So just a short thing about squares of opposition, affirmative, negative. So for each of the genetic code table, now we can calculate position of the unit interval. Of course, the general pattern would be that the that the ends and beginnings of the unit interval are A and U, and C and G are somewhere in between. As is theoretically and logically follows from the square of opposition. So but of course, we can ideal positions are 0 for U, 0.33 for C, 0.67 for G, and 1 for U, for A. It's basically 1,357. That doesn't matter. The XOR itself, which is in the core of Klein group algorithm, is actually a complementarity. We have true and false true functions, and the strong and weak groups are separated because C and G are separated from U and A, so the complements. And interestingly, it fits very well with the mutation ring, UCGA. You see the same patterns. Those are 4 tables compressed in 1. 1 and 2 means F is in 1 and 2, L is in 3 and 4. And the only joint column is, of course, identical for 4 is C column. Now we can calculate, of course, according to columns and rows. 1, 3, 5, 7, and Cayley table, which gives the permutation of all elements. So basically, we will see how this works when we put it in the algorithm for the weighting the differences in sequences with standard Mr. Bias and muscle. Okay, UPAC ambiguity code was applied. As I said, it's important for also analysis of some differences in sequences. And we have a situation, we see a central part of the algorithm, and so with 6 hydrogen bonds of diplets, and 4 bonds. Another pattern is for pure pyrimidine. And the third pattern is for amino and keto. Now, giving them 0, 1s, or we obtain clusters which can be put into multivariate analysis, and we obtain very good results in accordance to the probabilistic spheres I mentioned, and the calculations. And these are calculations of thermodynamic parameters correlated to a very simple model for possible for hydrogen bonds is given 0. 1 is given 6 hydrogen bonds, and the ones which are combination true-false is given 0.5. So it's a simple model with a regression of 0.98. These are original data for duplexes, and we will also use this algorithm to test the evolutionary trees, how they behave. So for the substitution matrices, we use Blossom. It's a standard method used today. Blossom matrix is used to get a sort of standardized phylogenetic tree, and we wanted to see, of course, how it works So these are results for REC-A in 3 kingdoms, and we see that it works very well. Eukaryota, bacteria, and archaea give 3 clusters. Gram-positive and gram-negative and mycobacteria are well separated, and all 3 kingdoms are well clustered. So these are, we have more of each species. However, it was very impractical, and nothing changed if you add some other organisms, regardless of in each of the kingdom. But it's visually not very easy to follow. So we put it into reduced it to 22. Now, columns, famous columns, which carry on the information about hydrophobicity and classic polarity. Its result is excellent. The same 3 clusters, gram-positive, negative, and mycobacteria. Archaeas are here, and eukaryota here for human and monkey, and you see the species. It's clustering practically the same as blossom. Hydrogen bonds, those 3 clusters out of 6, 5, and 4 groups. Also, excellent clustering. So bacteria, eukaryota, and archaea. Everything is on its place, so hydrogen bonds are surely important for the business. What one can notice is that the representation from the Klein 4-group elements, so from the group permutation algorithm, are in some senses even better present diversification within a tree. You see, it's not so close. But anyway, the final conclusion would be it's very similar. And all 3 clusters are separated. Now, this is the initial result of thermodynamic measurement by Shea of duplexes, and it also works well. So you have bacteria fine, gram-positive, gram-negative, mycobacteria as well. The same is, then we have archaea, eukaryota. Some other algorithms, K4, just a moment. For example, Cayley table. Cayley table gives all the permutation of the group within tables. It's a standard Cayley table which describes group permutation in general. Gives also a good result. However, there is one obstacle, and this is for all the first group was perfect. Columns and hydrogen bonds, whether theoretical or experimental. Now this is second group. It's Cayley tables, rows, and Rummer. It's also good. The only thing are mycobacteria, which are, of course, in the bacterial cluster. They have the same origin, but they are separated. Gram-positive and gram-negative work well. Those are rows. The same thing. So rows and Cayley table give the three kingdoms are separated. The mycobacteria are not put in the same branch, but they have probably the same ancestors because they, you see that. And then we have weak and strong group only. So only weak and strong. The same thing. The mycobacteria and have that difference, but the kingdoms are and all other elements fit in as they should. Monkeys with human and chicken and pig, horse and so on. Whom are practically the same thing. So they all work in a general way well. There are some variations, as I said, within a tree for some, and the others are identical to blossom. So the results indicate that different operator positions based on group theory permutations and not any ideology as well or measurements and group testings in Cayley table and its the rows or columns of the indicate diverse aspects of the cluster analysis. Column positions are mainly linked to the physicochemical parameters, while row positions, as I said, have some reflection of coevolution theory. The clustering base of Klein 4 groups is also successful as with hydrogen bonds. So it remains what do further. According to Marcello, during the evolution there is a phase of optimization rules, the ambiguity is eliminated and the system produces its components, and in the final end we have several genetic code tables. For each of these tables slight variations in group theory and indices can be done. However, so we could calculate and optimize particular transitions as evolutions for different genetic code tables on one point, and the other point is, of course, the point of that Julia said about tautology. Tautology is in this way avoided because we are looking at differences in sequences and scoring them according to unbiased recombination parameter based on pure or ring and group or ring and group changing principles, which have a fixed thermodynamic measurements, and we know that they are therefore so in this sense we have a chemical terminology or physical-chemical terminology tuned to group theory, and no selection of data set on which the matrix is based influences the result. So in that sense it would be a sort of continuation of this Kimura's King and Duke's theory of neutral evolution, with which we could calculate now, and of course compare it to the standard matrices. So to see what goes on, what are the differences, and this is just the beginning. These are preliminary data, but I think at this point it looks convincing. Thank you very much.